Hello children, I am Shubhajit. Welcome you all for watching a new and interesting video about the topic of cell division and cell cycle from the lesson Reproduction of Class 10 Biology. We know that all living organisms continue their species by reproduction. How does the process of reproduction take place? Is it not wondering that a parent cell divides to become two daughter cells? Yes, it is. Reproduction in unicellular organisms like amoeba occurs by binary fission. So, reproduction is a process of cell division. Have you observed that any wound with ruptured skin is replaced with new skin? This is also possible with cell division, and damaged cells are replaced with new cells. Now, we will have general idea that cell division is for producing new cells and also repairing the damaged cells. Learning outcomes. After studying this topic, you will be knowing about discovery of cell division in human beings, cell cycle, mitosis, meiosis, keywords, chromosomes, sister chromatids, centromere, interphase, mitosis, cytokinesis, meiosis. Let us see the concept map on cell division and cell cycle. Cell division and continuation of life. The cell division occurs in normal body cells and the sex cells or gametes. Virchow proposed cell theory and gave the phrase omnis cellula decellula. This means cell arise from pre-existing cells and indicates the importance of cell division in the creation of new cells. In 1852, a German scientist, Robert Remak, observed embryos and published his observation on cell division. He stated that binary fission of cells was the means of reproduction of animal cells. In 1879, Walter Fleming observed cells with string-like structures in the nucleus split longitudinally during cell division. He named such a process of division mitosis. Mitos means fine threads, as the dividing structures resembled threads. Later, these thread-like structures were named chromosomes, meaning colored bodies. Wilhelm Rox proposed that chromosomes carried a different set of heritable elements. Fleming observed the longitudinal splitting and the equal division of these elements. Gregor Mendel discovered the heritable elements in peas and approved the central role of the chromosomes carrying the genetic material. August Weismann hypothesized that individuals of the same species have the same number of chromosomes. In successive generations. In successive cell divisions, the number of chromosomes always remains constant. In 1904, Theodore Boveri confirmed the scheme of the mitotic division. In 1953, James Watson and Francis Crick determined the chemical nature of deoxyribonucleic acid or DNA. Mitosis takes place in all body cells which retain the same number of chromosomes. Meiotic division takes place in sex cells where the chromosome number becomes half. Cell division in human beings. The cell is the structural and functional unit of life. The process of cell division is highly evolved in human beings and is highly regulated in multicellular organisms. Cells in the heart and the brain never divide. The bone marrow cells actively divide to produce red blood cells. Wound or injury may bleed, and soon a blood clot forms to stop bleeding. This brings in various chemicals, stimulating the skin cells to divide and heal the wound. Cell division stops on the healing. But the cancer cells do not respond to growth regulating factors and divide continuously, ultimately killing the host. Cell cycle. The process of cell division is called mitosis and is completed in 40 to 60 minutes. The period between two cell division is called interphase. In this period, the genetic material makes its copy and is equally distributed to the daughter cells. Interphase can be divided into three phases. G1 phase or gap 1 phase is the linking period between the completion of mitosis and the beginning of DNA replication. The cell size increases during this period. The S phase or synthesis phase is the period of DNA synthesis and duplication of chromosomes. The G2 phase or gap 2 phase is the time between the end of DNA replication and the beginning of mitosis. Cell organelles divide and prepare chromosomes for mitosis. The M phase is the mitotic cell division phase. Johnson and Pottu Narasimha Rao revealed the structure of interphase chromosomes with the cell fusion technique. These cells contain a nucleus and chromosomes. Before any type of cell division, the chromosome number is doubled. That is, if chromosome number is 2n, it is doubled up to 4n. Chromosomes are structures made of DNA and proteins. In humans, there are 23 pairs of chromosomes in each cell. The structure of a chromosome can be divided into two main parts, the centromere and the arms. The centromere is the central region where the two arms meet. It plays a crucial role in cell division and is responsible for pulling the chromosome apart during mitosis and meiosis. The arms of a chromosome can be the P arm or short arm and the Q arm or long arm. The arms are composed of DNA and proteins tightly coiled to form a structure called chromatin, giving the characteristic X shape to chromosomes. Now that we have covered interphase, chromosome structure, so let us move on to the mitotic phase, the exciting part where cells divide. Somatic cells and stem cells divide by mitosis. Mitosis is completed through two main steps. Those two steps are karyokinesis or nuclear division and cytokinesis or cytoplasmic division. Karyokinesis is completed through four steps. The first stage of mitosis is prophase. In prophase, condensation of thin thread-like chromosomes occurs and they become short and thick. The chromosomes appear with their pairs of sister chromatids. The centrioles duplicate and move to opposite poles of the cells. In this stage, the nuclear envelope breaks down and the spindle apparatus forms to guide the movement of the chromosomes. In metaphase, the chromosomes line up along the cell's equator to get evenly distributed into the two daughter cells. Then comes anaphase. The sister chromatids separate and move towards opposite ends of the cell, ensuring each daughter cell receives a complete set of chromosomes. Lastly, telophase marks the final stage of mitosis, where two nuclei form around the separated chromosomes. In this way, karyokinesis completes and cytokinesis begins. The cytoplasm divides by cytokinesis and two new cells are formed, called daughter cells. A notch or groove is formed at the equatorial plate of the cell. This deepens gradually, forming two new cells. However, in plant cells, instead of the notch, a cell plate is formed exactly along the midline of the cell. The cell plate continues to grow outward until it reaches the cell walls on either side and fuses with the existing cell wall. Thus, cytokinesis is completed. Mitosis is necessary for the growth of the body, wound healing, and formation of blood cells, etc. Unlike mitosis, which is a continuous process for cell division in most cells, meiosis occurs only during the formation of gametes in sexual reproduction. Meiosis has two phases. In meiosis 1, the cell's DNA replicates and the homologous chromosomes pair up, forming structures called tetrads. Tetrads play a significant role in exchanging genetic material between maternal and paternal chromosomes through a process called crossing over. This remarkable phenomenon introduces new combinations of genes, ensuring genetic diversity in offspring. Meiosis 2 is just like mitosis because the two haploid daughter cells formed in meiosis 1. These undergo division by the separation of recombined sister chromatids, forming 4 haploid daughter cells N from one diploid 2N cell. Thus, meiosis is also called the reduction division. The process of gamete or spore formation occurs through meiosis. During meiosis, crossing over occurs, resulting in genetic recombination. Due to this, all 4 daughter cells are genetically different from the parent cell and each other. Further, you learn elaborately about cell cycle and cell division in higher classes. Hope you like this video.